So, we've been able to see why we need proteomics. We've discussed how we can prepare our samples. We've been able to see how we can get an overall picture just using some different ways of electrophoresis separation of our proteins. And we've been able to see how we can detect separate single proteins or group of proteins with the use of immunoassay methods like Western blotting, like ELISA. But since we are more interested in proteomics and being able to see the whole picture on different proteins, different modification of proteins and so on, we need a little bit different methods. So up to date, we've only discussed methods of, let's say, protein chemistry. And today we'll start with methods which will allow us to see the picture, not whole picture, of course. We can't see every single type of protein which is in the cell. For instance, some proteins are only at low-level copies, like 10 copies per cell. There is no methods that could provide us with such a sensitivity just to be able to quantify those 10 proteins. We can actually qualitatively say that there are such proteins with some additional histochemistry. For instance, if you are in microscopy, you know that there are some methods which allow you to detect even single interaction between antibody and antigen, and you can then amplify it some million times to see that there was such a protein at least. But of course those methods are prone to errors, prone to noise and so on and so on. So usually we can only detect proteins from some concentration, not lower than some concentration. And one of the methods that we'll be discussing, we'll start actually from discussing, is a method which you probably know from different courses, which is the use of microchips. You can use some glass or some other substrate which is specially designed to bind to proteins. You've already done so with, let's say, nucleic acids. You can activate the glazed surface with some chemical agents, and then those chemical agents will covalently bind either nucleic acid, if you are interested in, let's say, sequencing, Illumina sequencing is one of the examples, or you can modify this the same surface that it can bind to some proteins, some NH2 groups, some other groups. So the proteins of interest will be covalently bind to your surface, be it glass or any other surface. And then you can analyze those proteins. We've already seen this in ELISA methods, right? Just the same, just surface was some plastic and some wells of microwell plate. But with this method we can probe a lot of proteins simultaneously. Like we have some solution. This is, let's say, self-made way to produce such microchips. As you can see, there's just a 3D printer which have some special holder for pins. Those pins are put in solution and then just make some prints on your slides. Then you can get another set of pins, another set of solution, and so you can print such kind of microchips by yourself. Afterwards, we can use the same technique as was used in ELISA. We can either fix an antibody which will capture the protein of interest, or we can bind something that will bind any protein that is here and then probe it with antibody. So anything that is in ELISA is also possible. So I encourage you to see a lot of different methods. There's a plethora of different methods of ELISA, how you can catch your sample, how you can detect it, how you can even transfer it to some other solution. So a lot of different ways, but in the end we'll be able to see, using just conventional fluorescent microscope, for instance, picture like this one. Every dot is this point where we deposited our solution, and if we do some kind of laser-like protein capture and detection, every dot here has some intensity, and this intensity after calibration just like with ELISA, can be counted as a copy number of protein or concentration of this protein. Of course, copy number is not actually feasible. We cannot detect really small concentrations, but let's say concentration of a protein in your sample, right? So at the same time, you can probe your sample for a lot of different proteins. So the problem with ELISA and Western blot was that we can see only a small set of proteins. Like if you even try to do some Western blotting with fluorescent labels, you can label simultaneously up to 4, I think, different proteins if they are in the same band. Of course, if you make some different separation, which allows you to separate your protein better, you can label different proteins with the same label and know what is this protein, just the position in the gel, right? So actually it's possible to make more than 4, but it's troublesome to do. So this method allows to see, as you can see, a lot of different proteins at the same time and even get some quantitative information, how much protein was there. And as you can see, it's possible to produce such chips by yourself. What is even more? Yep.
May I ask about
Of course.
how we calibrate this information? Just visual analyzing, I mean, or we have some numbers from this picture?
You will get numbers of intensity. You have a microscope with a camera. Camera is calibrated to have the same count of intensity to the whole field of view. So whatever it would be in the center of your field of view or somewhere in a corner, it will be calibrated. It will have the same amount of intensity counts. Like if you have 8-bit camera, it would be from 0 to 255. If you have 20-bit camera, it would be higher. And now you've calibrated only that your instrument is able to measure this intensity at the center of the field and at some position. And you need to calibrate your chip because you can actually have a little bit different concentration of your solutions here, for instance, and so on. So you can input here something that will be able to bind to some protein which is not in your sample, which you add externally. And then you can see whether it binds and what is the intensity of this binding here. And so you will be able to calibrate your sample. It could be used even as an internal standard in a separation procedure. So you could start by adding this protein in your mixture before you start even your sample preparation. And this protein will be lost as much as all the other proteins in your sample, right? So you can calibrate what was the actual concentration of your proteins compared to this known standard of known concentration, because you added it at the early stage of protein extraction, separation, and so on. So it can be calibrated, but also what is quite interesting about this method, you not only can put here antibodies. You can even put some other proteins. For instance, if you are interested in protein to protein or protein to nucleic acid interaction, you can put on this microchip some protein which interact with other protein, and you can see this amount of interaction between them because those proteins can be made immobile on this surface. And then you can stain it and see what is the intensity of those dots. So this method either allow you to identify this protein if a known kind of antibodies were used, or you can even see what kind of different proteins can interact with your protein. For instance, you can separate different proteins from your cell. You can put it on such a slide, and then you can try to ask question whether my protein of interest can interact with any of those proteins. You just put the solution of your protein of interest here, then you can wash it away, and to make sure that those complexes, if there are complexes between different proteins, stay there, you can fix them by means of some chemical agents like formaldehyde, for instance. It will commonly bind those proteins. So even if complex was not quite stable, but was there at the time of applying this fixative, it will stay there forever. Then you can add antibodies and see what kind of proteins were able to interact with your proteins of interest, right? So quite an interesting technology, not quite widely used because it was superseded by some other methods, mostly with the use of mass spectrometry. We will be talking about it a bit later, but still useful. Also, there is another variant of this method, which is used in clinical environment mostly, is the idea that we can actually put our antibodies or any other proteins to search for other proteins on a small plastic beads. And then use something that is comparable to cell sorting or fluorescent cell activated sorting machine. Have you had any course on such a machine which allows you to see different cells in solution, count them?
Like
Flow
So yeah, flow cytometry, right. So, but here we have not cells, but those small plastic beads. And what we can do actually is that we can produce a lot of different beads, which will differ by size. And also, let's just produce 9 of them. And also by some fluorescent signal, which is embedded in those beads. Let's say there would be a lot of such signal, there would be a little bit, and here will be just some signal. Then you can use really small, really simple kind of such machine, which will have a stream of liquid with your beads, and after having a certain just size and fluorescent label, you can actually get a graph of your beads here. What can be done next? You can activate those beads and put some antibodies on those beads. Let's say for this bead we'll put one antibody to one certain protein. On this bead we'll put another antibody to another protein, and so on and so forth. But we will then just stain either protein or antibody. We can do it both ways. Then we can add another channel here. For instance, those beads will fluoresce in near infrared when we illuminate with red light. And we can add here something that will fluoresce green when it's activated with blue light, for instance. And here you know by this table which kind of bead is where, and you know what kind of bead was labeled with what antibody. So, in fact, it's a multiplexing of Western blot analyzers which can be done in solution. You only need your protein of interest or mixture of proteins like human serum, for instance. You can just mix it with those beads. There are protocols how to stain, how to do all this work, and then analyze it, and the machine automatically can classify all those beads here, like this, and for each class of bead, it will get you some intensity. Of course, you can calibrate this machine, no problem. If there is some known standard, you can get just solution of known concentrations, get it to this machine, and you can calibrate it even quantitatively, or semi-quantitatively actually. So was it understandable how such a machine works?
Yeah, I ask some details because it's not clear for me actually. What is the sense of different size? As I understand right, we put all such types in
one mixture.
In one mixture.
Yeah.
So we have different size of such balls and different
signals. Yeah.
Different antibodies, but in the same time, the same protein can be linked with different antibodies. antibodies?
If I get you right, the question is how we can have a relation between the class of our bead and antibodies which we put on it, right?
Yes.
Probably. But it is also.
I can ask. I can actually make some. I can ask in Russian. We need to do.
Yeah, I understand the question,
I
but it is also a good question. But I mean something else. But it is also.
Okay, actually, we can make those communication with an antibody and different classes of those beads even by ourselves. Those beads can be sold as a set of, let's say, 9 beads. Each bead will be in its own vial, for instance. So one vial will contain only this class of beads, another vial only this class, and so on, so on, so on. And there are chemistries which allow us to bind proteins on those beads covalently. So we just add here some, let's say, green antibody. Here we'll just, for the sake of understanding, put here some red antibodies, and then we'll have in solution all the beads of this class labeled with, let's say, red antibodies, all the beads of this class, for instance, green antibodies, and so on and so forth.
Can I ask?
Okay.
I think I understand your question, Shipment. Why there are small beads and large beads? What would be the differences?
That's a really interesting question, and absolutely.
Variety.
Yeah. If you use only the intensity of fluorescent signal, you can get like 16 different sizes. If you get more differences in intensity, it wouldn't be possible to separate them as good as here. As you can see, even for one class here, there is some dispersion of this intensity, right? And if you will have a lot of different classes which differ only by fluorescence, you will mix them. You will not be able to say that this is this certain class, for instance, right? And that's why we need another dimension. We could either have different, for instance, let it be blue, different fluorescence signal, and we can multiply. So we have 3 classes of, let's say, intensity in red channel, and we can have also 3 classes in, let's say, blue channel, and now we have 9 different variations. If we have 10 and 10, we have 100, and so on, so on, so on. But size is really easy to detect. You need somehow to detect that there is such a bit, right, to start analyzing. And it is possible to see the size of this bit just by the properties how it disperses light. For instance, if you have a really tiny, small speck of dust in some, let's say, we have our projector, then there is a little bit of dust in the air. The smaller the dust, the more would be, the more it will disperse light in different directions. Yeah, so the angle at which the light will be dispersed will be bigger, and you can just easily detect it. Like if you have, for instance, let me put here an idea. You have some capillary, there is some focused bits here, and you have some, let's say, laser light there. If the bit is small enough, then there will be not so much, there will be much of dispersion in this direction. If the bit is bigger, the dispersion will be smaller. So what you can do here, you can block the light which can pass through when there is no bit, and put a detector here which can get the intensity of this light. The detector will be big enough, so if there is a really small bit, we will have only some light here, the angle will be low. If you have something that is quite small, it will be highly dispersed and so on, so on. So it's just a way of multiplexing different types of your beads. You can actually make it even in a 3D. You can add another channel of fluorescence and size, or you can make those beads of different internal structure, for instance, like in cell sorting machine you have this side scatter and forward scatter, for instance, right? So you can use different detectable characteristics of those beads to be able to classify them. And then when you know the class of those beads, and you need another channel to detect how many of those proteins were able to bind to this bead, right?
But I still don't understand why the different antibodies can, in this case, are linked with the same protein.
Green protein.
You made it so, or you buy it, such as said, because what you can buy, you can buy.
What is the
vials.
sense of this type of antibodies? Different antibodies
For different proteins. For
But-
instance, you have a serum.
Yeah,
Yep.
it's okay, but in this picture it looks like the same proteins are
The same antibodies on this class of beads. On another class of beads, you have an antibody to different proteins.
So these green proteins on black and green antibodies, they are different proteins.
Right.
Ah,
So each class,
okay. They just looks like the same protein because of
but
color.
they are differently colored. Right?
Proteins are not.
I
Proteins.
see. My bad.
So, and
of
it
course,
was
yeah.
confusing for me.
It was just some mishap. I wasn't able to, yeah.
Okay.
That's the right idea here, actually. And let's put.
Okay.
So the idea is that you can now do multiplex Western blotting or ELISA, whatever you want, in the same solution of multiple different proteins. It is understandable why medics like this machine, because you can just put some human serum here and then just go analyzing what was there and whether there was some increase or decrease of some kind of proteins, right? But in fact, for us, it's just a multiplexing. Because it's not the proteomics per se, but it's already close to this because we can analyze not single protein, not just set of some small number of proteins, but a lot of proteins at the same time. So it's already more close to proteomics. And since we can use antibodies or even monoclonal antibodies, we can sense not only proteins, but also their surgery structure, for instance, their post-translation modification. Whatever can be detected with monoclonal antibodies can be detected with such a machine. But the same is true with microchips as well. And they can be produced in-house by yourself, actually. But still. The problem is that for such kind of detection, we can do just, let's say, less than 100 different proteins, which is a problem. Because our proteome, as we've already been talking, is at least more than 1,100 different proteoforms in cell, right? And we can't multiply it, so we can't get antibodies to every possible post-translation modification, every possible proteoform. It's just impossible to do. It would cost enormously, and well, it's doable, but nobody will try to do so. And what we can actually do, we can first separate all the proteins, at least more proteins than can be analyzed with such a machine, and then label it, even as we've done with Western blot, or identify those proteins by the means of mass spectrometry, which will be the topic of today's lecture, actually. And to do so, we need to separate our mixture of proteins by two different characteristics. For instance, isoelectric point and molecular weight. We've already seen those 2D gels of different ways, which allow us to get more information, but if you've seen, those gels were of a low resolution, right? And were able to solve only some kind of problem, like complexes of proteins, for instance. And 2D SDS page gels with isoelectric focusing can actually allow us to resolve more than 1,000 proteins, even up to 2,000 and some publications. So we already can get a lot of different proteins, and I will show you how you can get even better resolution. Of course, it will take some time, but it is possible. But for such a 2D gel to be able to actually make and to get it, we need two different environments. First, the problem is that to do isoelectric focusing, we need special conditions, and any salts in buffer, any problems with like say denaturing agents which have its internal charge will make this focusing impossible, right? Because if we have protein and want to know its isoelectric point, if we add some SDS on it, it will have different isoelectric point, because SDS will also bring some charge. And here comes this nice molecule which was first used, but there are actually other molecules. As you can see, it acts as SDS, but there are two different groups, and net charge of this molecule is 0. But it can allow us to solubilize our proteins. So the problem, if we have proteins in solution, some of them are insoluble. Some of them will become insoluble if concentration will be high enough of these proteins. And if you want to actually make this isoelectric focusing, what we'll do, we'll increase local concentration of those proteins in our gel, right? And some of them will just become insoluble. So we need some agent which allow us to solubilize these proteins, like SDS or something, and this molecule will serve as such a surfactant which allows to solubilize our protein, but we will not in any way change its isoelectric point. That's why it's a really interesting idea. And we've already discussed what is isoelectric point of a protein at certain pH. This protein will have 0 net charge. And what is different from the electrophoresis that we've discussed last time is that this electrophoresis is an endpoint electrophoresis. If you will just continue to run it, it will stand at some parts of this gel and will not move anywhere. If you will wait a little bit longer with conventional electrophoresis, all your proteins will just go from a gel into solution, right? This is some endpoint when it will be focused like this and you can use it afterwards. So, since we have such an agent which allows us to solubilize our protein, we can immobilize also pH gradient on such plastic pieces of plastic, and now we can use electrophoresis, which allows to create a gradient of electric field, which then moves our proteins to its isoelectric points. And we have separation in the first dimension. What we should avoid? To be able to focus such proteins, we need to create really strong electric gradient. So there are some kilovolts of voltage. And if our sample will contain some salts, what do you think? Will we be able to create such a big gradient of voltage on our strip of gal without any consequences?
Looks like no.
No? Why?
But this
The salt, the going gray, would make acidity-basic reaction.
Actually, even easier. The problem is that the salt is really good charge carrier. It will, in fact, just short all your cell and will provide some way for this current to flow. So you would have not only some voltage but also high current. And high current and high voltage will provide you a lot of Joule heating. Your cell will just overheat and actually boiled gels are really not quite good for protein separation. So the main idea here, if you will plan to do that to gels, you have to get rid of any salts in your solution or keep it at some 10 millimolar or even smaller than 10 millimolar level to be able to withstand such a different gradient of voltage. And also you need to be sure that no conductive substances aside from those salts are there. After that, you also need to be sure that there are some agents which will make your protein soluble, like CHAPS or Triton 100. It's also possible to use it as a substitute in our conditions, because CHAPS is a little bit pricey chemical, but it can be substituted for some low quality work with Triton 100, which is, I think, available in any laboratory. And what we have now is this strip of a gel which is focused with our proteins. We can put it on top of an SDS page gel, and just to make sure that this strip wouldn't move anywhere, we need to add here some agarose. And then we can start our second dimension, just SDS page gel, which we've discussed previously, and get this separation. But there are some points that I have to stress, is that, first of all, what do you think? We have pretty mild conditions which were used to separate our proteins. We haven't had any SDS here or anything, and since there was no harsh condition which can restore some covalent bonds which can form in our proteins with cysteines, will those proteins migrate at the same rate in SDS page? For instance, if we have fully gelatinized protein, or protein which still contains some disulfide bonds in it, which will migrate faster. The same amino acid sequence, but different.
The second.
The second one, right? Just like we have this idea of runners through the forest, right? If the runner can just bend his stick and he can run faster through this forest, right? So the same. So to be able to make this 2D electrophoresis, we have to somehow get rid of those cysteine covalent bonds, and we can add here two chemicals. Actually, when we were discussing SDS page, we've talked about mercaptanol as one of such chemicals which can disrupt those disulfide bonds, but mercaptanol is not good for 2D gels for some reasons, and we have to use different chemicals. One of them is dithiothietol, DDT, which breaks those bonds. But what can happen if we'll place our proteins, even denaturation with some cysteines here, in solution at room temperature for some time?
At which temperature?
At room temperature.
It can
DDT. Yeah, absolutely. Some bonds can again form. So what we can do? We can actually make this SH to S something like acetylation, for instance. And we need to use some agent which will modify our protein, so we can do some post-translational modification by ourselves to prevent reformation of those disulfide bridges. Because in this case it can lead to those bridges. In this case, if we have some acyl here, and even if there is no acyl, no bond will form here, right? So we can reduce this with DDT and add iode acetamide as an agent which provides with such an acetylation. So now we have all the proteins which are here, but of course there are some modification of cysteines there, and we can get second dimension which will reflect molecular weight of this protein, right? or actually its length, to be sure. And such gels, we'll get to this later, will look something like this. As you can see, we can resolve a lot more proteoforms here. Even for some protein, there is a charge series, so-called. You can see that there is a protein of high molecular weights, let's say 200 kilodalton, and there are a series of different proteoforms of the same protein. The charge is different. For instance, there is some cases of phosphorylation, for instance, right? So isoelectric point will be different, but the mass will be the same. So you can see not only proteins here, but also proteoforms. For some cases it's good, for some cases it can be detrimental. For instance, if we use some protease inhibitor as ABC, which is actually irreversible, it binds to a protein, it itself can add some charge. So some proteoforms here can be an artifact, but still you can resolve a lot of them. I have to get back to this slide just to describe how can we make such a gradients of pH and how we can immobilize such a gradient. For this, we need to use molecules which have some certain charge and some certain pH, and which can be copolymerized with our acrylamide. So we can put those different molecules in solution. As you can see, there is a lot of different pHs at which those molecules will have no net charge. So we put those molecules in solution. We add here some acrylamide, we can add even bisacrylamide to them, and then we start the same isoelectric focusing without any proteins. And those molecules will also be isoelectric focused in, let's say, solution, gel, or whatever. And then we can add agents to polymerize this gel. Afterwards, as you can see, they all have this motif which allows it to be polymerized, copolymerized with our gel, which is based on acrylamide, and you will get those immobilized pH gradient because they themselves will provide this pH. They will be inside our gel, and so we can get this gel with immobilized pH gradient. It's not for remembering, it's just an idea how it can be done. And those molecules are called ampholites. Why I have to put it here? Because at some point of your work with proteomics, it could be beneficial for you to do preparative isoelectric focusing. For instance, you have a mixture of different proteins, but you are interested only in some class of those proteins. And those proteins have the same mass of some different proteins with different isoelectric point. And those different proteins are at really high concentration. You could either use some antibody-based assays to get rid of those proteins of high concentration, but it would cost a lot of time. It's easier to make isoelectric focusing in preparative way, and then just to get this fraction of proteins which have those pi, and then use only this fraction for even 2D gel or 1D gel or Western blot or whatever, or mass spectrometry. Anything can be possible. But there is one thing. As you can see, those molecules have some remembrance to amino acids, right? Charge, some groups, and so on. And if you will have those ampholites in solution when you will do your 2D gels, like this picture here, those ampholites will be dispersed through your cal, and they will be also colored by Kumasi stain, by Orel stain, by silver stain. So your gel would look like it has some haze on it. So everything will be stained. So if you do such a preparative work first, you somehow have to get rid of those ampholites. You have to wash your gel or have any other protocol to get rid of those ampholites. If they are immobilized in the CGP stream, no need for additional washing because they will stay in this gel. So now what is the take-home message here? That doing those 2D gel electrophoresis is really a good way to get more information, and now this is already proteomics. We can distinguish hundreds and thousands of different proteoforms. Of course, it's not a whole proteome, but a big part of it. And what you can do, you could actually compare two different gels side by side. For instance, you are interested in what changes in the proteome of your organism of interest and different conditions, be it control group, diseased group, or you just have some heat shock for instance, E. coli, and you can see what would be the difference in proteomes of those two samples, just comparing two gels. And then you can get points which differently synthesized or expressed. It's possible to say in English that proteins are expressed, which is a little bit strange in Russian, but still. And then you can excise those parts of gel and identify what were the proteins that were differently expressed. In English, they are usually abbreviated as DEP, differently expressed proteins. So if you came by this abbreviation in some articles, it's just the proteins which differs between two proteomes, like in this case. So the first take-home message is why we need those two gels and how can we use them. The second take-home message is that there are some methodological problems which we have to be careful of, is that we have to somehow get rid of all salts, that we have to use different solubilization agents like CHAPS or Triton X100, X100, and also that we need to be careful at this stage. Before we put it on our SDS page gel, we need to use those two chemicals in a special protocol to reduce those disulfide bonds and to modify our cysteines to be able to resolve molecular mass or actual the length of our proteoforms, right? Also, what do you think? Since there is already some certain gel in which our proteins are embedded at this first stage of isoelectrofocusing, do we need some special staking gel here? No, there is no need for staking gel because we embed this part of gel just in agarose to fix it. There would be already discontinuity in our system, so they will be focused by themselves. Nothing needed to be added here, and we'll get, hopefully, something like this. If you will be doing this by yourself, of course, there are some protocols which includes steps on how to rehydrate your fixed pH gradient gels because they are shipped in dry conditions. Pay attention how to do this because there are three different ways on how to do this, and it will actually, the way how you will rehydrate them depends on what is molecular mass of most of proteins of your interest. Just if you're interested in some low molecular mass, middle molecular mass, or high molecular mass, three different ways of rehydrating these gels, which can actually have some impact of your results. Anyone plans to use something like this in your thesis work? So, then everything can be seen on YouTube or read in article just to make sure. What actually also really nice about those two gels, that everything that we've been discussed up to the date, like Western blotting, like having some different ways to stain those gels, is also applicable here. For instance, we can use specialty dyes, which can be used to see post-translational modification, like phosphoproteins, like glycoproteins, and then we can stain our gel with just chromatography to see all proteins at all. And we not only see the proteins themselves, but also differences in post-translation modification, because, as we've already discussed, for some cases it's not the quantity of protein which actually differs, but post-translation modifications or turnover. Turnover, of course, it's not possible to visualize with this technique, but post-translation modifications you can see, at least for glycosylation and phosphorylation, are quite possible to be distinguished, recorded, and used.
May I ask a question?
Yeah,
Maybe
certainly.
how we understand, maybe I somehow didn't understand this, how we understand which certain post-translation modification is on the gel. So,
There are specialty stains which will stain only, like say, serine which have phosphorylation on them. Like in histology, there are stains which will stain only certain substances. Most of such stains are fluorescent stains, so you don't need a high concentration of this stain. And since it's since it's a stain that uses fluorescence, at it can be at different wavelengths. You can actually image those two modifications at the same time, just using different excitation light and registering different light from your gel, just by wavelengths.
but can we understand the certain position like on this?
Absolutely, absolutely. You can stain either you can have some marks on your gel, like follow the gel or something, and you can just open, for instance, Photoshop and put different layers one on top of another. And then you can see that this point here was stained, let's say this group here was stained by Kumasi, so there are at least proteins here, also we hope. And there are two dots here which are stained by phosphoprotein, staining fluorochrome, and they are phosphorylated. So there are different states of phosphorylation because it's the same molecular weight. You can say that probably it's the same protein, right? And also there is another dot here which is a little bit higher because glycosylation will add some mass to your protein, right? And also it have some additional charge here. So probably it's some proteoform which have additional glycosylation with some group which changes its charge also. So probably it's the same protein and three different modifications. And if in one case one of these modifications is missing, then probably the difference between two protons is just this combination of post-translational modifications. Was it clear, or do I need somehow clarify this idea? Like in fluorescent microscopy, you can stain, well, you can't use word stain for fluorescent dyes label. Let's say you label your cells or your tissue with different fluorescently labeled antibodies. You can actually at the same time get three different wavelengths, three different pictures, overlay them and see whether there are some cases of colocalization, whether there are some like multi-channel fluorescent images. Everyone seen them at least in some articles, publications, right? So the same idea is here. Also, since we haven't yet fixed our gel, we can do immunoblot here. Nobody says that it's impossible to transfer such a gel to a membrane. Then we can actually ask whether this protein is some protein which we have antibodies for recognition of this protein, right? So it's already some kind of proteomics. Of course, the problem here still stays that the resolution of this method, using some mini gels of some several centimeters, is not good enough. And what we can do, we could either have bigger gels, but if somebody already worked with gels, then it is obvious that working with big gels is troublesome, at least. They break, it's impossible to reproduce them in the same way, or they will have a little bit different pictures, so on, so on, so on. But what we can do actually is that we can use different gradients of pH, of immobilized pH here, to be able to zoom in some certain region. We'll have the same small 7 centimeter gel, but in first dimension we have only proteins, let's say from pH 4 to pH 6, for instance. And of course we'll be able to resolve a lot more proteins or proteoforms here, and we can even make some longer gels to resolve even better some molecular weight here. So it's not always needed to make big gels to get big resolution, because as you can probably remember from our first lecture, the distribution of isoelectric points in most proteins, I'm talking about mostly animals, vertebrate animals, is something like this. There is 7.4, there is 6 point something, there is 8 point something. So we can, if our proteins of interest fall in this range, we can just get our strip of immobilized gradient here, or if there are some special cases of, let's say we have some histones, we can get something up to around here, right? And those different exactly focusing strips are commercially available, so you can just pick your range and you can buy it easily. Like here, you can make a big gel and have some resolution, or you can get those smaller parts and get even higher resolution. As you can see here, those proteins are not resolved. It's just one point. Here we have at least 3 different proteoforms. So, was it clear, understandable about 2D gels?
Yes.
Alright, let's then continue. For instance, we've got 2D gels, 2 of them actually, which comprises 2 different states of human patients, actually the same patient, which one of them is from diseased tissue and another is from, let's say, control or normal tissue. As you can see, even at these pictures, those gels were first stained, that's why they are black and our proteins are white here, but it's possible to compare them, and even without any software, we can see that this group of proteins is missing here, right? But it's not so easy to do it by hand or just by eye. Probably we would like to do something like this. Put those proteins one on top of another with those gels one on top of another, differently colored, like red and green. And of course, if we have some colocalization, we'll have yellow color, right? But as you can see, if we just put one on another, there would be such cases when the same protein in different runs of our gel will be at a little bit different position. Why so? Because proteins are also charged. If there's a little bit more of some proteins, it will, in fact, by itself, will just move other proteins at different positions during the electrophoresis. So it's not quite reproducible. And we have 2 different solutions to this problem. First solution, and I will show it on practice this Friday, is to use some special algorithm with the use of free program Fiji, and there is such a plugin, BNRMG, which allows you to just bend the gels a little bit in such a way that the difference will be as minimal as possible, because most of the proteins, especially housekeeping proteins, will be the same, right? And most of those points should actually have the same position. and you can get one of the gels, say that your master gel, and then align all other gels of the gel with the use of some software. First I will show you how to do this. And on this aligned picture, you can see the differences. There are some proteins which are red, which are present only in one state. There are some proteins which are green, which are only present in another state, and some yellow ones. But what do you think? Is it possible to quantify those differences from those gels? Quantify.
Differences is like the
Like say that
different proteins or I'm not sure.
differences in concentration.
Different.
We can say that here is the first one is more concentrated. Here is the second one more concentrated. But is it possible to get some numbers?
No.
No.
No, why? A different sample, and we need somehow to calibrate. We need the third one, which we mix together. Let's say 50% of the first sample, 50% for the second sample. Make this master gel, which have all those points from both diseased and controlled tissue, and then we have to align on this. How to say it? This gel, let it be gel. And after that, we can actually then compare them. We know that this master gel have 50% of first and 50% of second. And if something is more than this master gel, then one of them have more concentration. If less than this, the concentration is less than this some number, right? So, to be able to quantify this information, we need this additional sample, so-called pooled sample. For instance, if you have 10 different probes of the same organism, the same tissue, but at different states, you also can pool those 10 different samples together to get this master gel, which will, by definition, contain all those proteoforms. It could be that some concentration of some proteoforms would be low enough, but for most of the cases, we can use this master gel for alignment. But there are also some problems. This is how this alignment looks for one of the gel. As you can see, the more the length of an arrow, the more there is an error in the alignment. And what we can actually do for some cases is this scenario, unfortunately. As you can see, using this elastic deformation, we can align two different objects which are absolutely different, but they would look almost the same. So, with this approach, there are a number of drawbacks, but it can be easily done. And another approach is to pre-label our proteins of interest. If we have any ways to label our protein in a mixture, and we'll have, let's say, for instance, label of our protein from a normal tissue, be it some green fluorochrome, and we have our diseased tissue which is labeled by red fluorochrome. We can mix it together, and then we can separate them together using this 2D gel, and there would be no differences in their position, right? But there is a number of conditions. Those labeling shouldn't change isoelectric point of this protein, shouldn't change a lot molecular weight for our protein, and this label shouldn't be dense. It shouldn't label every amino acid, because after that we wouldn't be able to continue with our SDS page electrophoresis. It will change the characteristic of this protein and so on, right? Fortunately, there are such dyes which can have the same isoelectric point, which have different excitation and emission wavelengths, but at the same time they have the same molecular weight, or almost the same, and the same isoelectric point. And also we can label our protein at some minimal amount, only 3%, which will not change our protein characteristics much. But then we can mix those two proteins which are already pre-labeled, make all that we've done with 2D electrophoresis, and get this gel just in instance. We could imagine it with different sets of filters, and each point will, with the same proteoforms, will have the same position in gel. No need for alignment, right? So, quite nice, but using only two proteins or two samples without any additional measures, can we actually say that this red is the one that is bigger in concentration in the first sample and this green is something that is bigger in the second sample? Or do we need something?
To some extent, you can say.
But what do you think? Those fluorescent, right? Those fluorescent stains, are they have the, is it true that they have the same efficiency and that this red stain will be as bright as a green stain?
No.
No, so we need to calibrate them, right?
Mhm.
And it can depend on external environmental conditions. What we can do actually, we can add third label. Let's say, let it be, for instance, blue, but it's actually orange. And then we'll have three different labels. One is our control sample, another one is our diseased or our research sample, and the third one is our pooled standard, as we've already done with our 2D gels before, right? And now we can have some numerical information about the concentrations of our proteins, right? So we actually need this third sample of pooled mix to be able to work with it. And my question is, if you have more than two different conditions and that you have to compare, what will you do? We have only three different fluorescent dyes which can be used. So on one gel we can have only three channels.
Can we mix?
Pull all the samples together,
Yes.
right, and in equi-concentrational manner, and use this standard in every gel that we do for comparing two samples of this big set of samples, right? And then we can actually do the same that we've already done. We can use those control channels to align those gels, right? Because we know that it is the same sample. We pull them. And then every channel is what we compare in every case. And we can actually get now the means of comparing a lot of different proteins. For instance, we have the same plant, and we can compare it when it has some condition where there is not enough water, there is a lot of salinity, there is not enough light, there is a lot of light, whatever, and see what actually differs in those conditions. Because, for instance, in plants there is a lot of proteins which respond to any abiotic or abiotic stress, and they will be elevated in any conditions which are not favorable. Are we interested in them? Or are we interested more in some biomarkers of some conditions which are different from other conditions, right? That's why for some cases we need to compare a lot of proteins to be able to answer to our biological equation. So, that was it. Actually, you can see the chemical structure of these dyes. If you've already worked with either microbiology or microscopy, you can be familiar with derivatives of those dyes. CY dyes, they were patented by Amersham and by GE company. I hope that now this patent is not valid already. Really nice dyes, they are bright. The only problem with those dyes, which I have to stress here, they easily degraded by ozone. So if you have some printer, laser printer, copy machine or anything in laboratory, keep it away from those dyes. They will be quenched in no time. Just they have different excitation and emission properties, so you can actually separate them with the use of special filtering. And again, we need to do this minimal labeling. Only 3% of all lysines should be labeled. Actually, it labels proteins by NH2 group, and it can be either on N-terminus of our protein, but there's only one, and it's not enough to label the protein, or on lysines. And as you can see here, this group, which actually makes a covalent bond to NH2 groups, and those labels are proteins by covalently binding to them. So that is an idea that we've been discussing on how to be able to compare a lot of different proteomes. And if you have any questions about gel-based methods, please ask them, because we've fortunately done with all this gel wet laboratory stuff and can get to some more interesting part of our lecture. Alright, if no questions, let's continue. Then, this will be our third lecture. And we have now to discuss how we can identify the sequence of our proteins. Because up to date, we can only identify our proteins by using specialized antibodies, right? Which is not always feasible, which can cost a lot, which have some errors in it, and so on and so on. And we need some means to either fully sequence our proteins or get some information which we can use to get the structure from genomics or transcriptomics database. Because if even the sequence is not fully deciphered and we have only some parts of the sequence, we can search the database and see whether this certain, even not complete sequence can be assigned to some transcripts or some parts of genome. So we do not actually need to sequence proteins right now. We need to have some small information which allows us to search this database for sequenced organisms. But we'll have to start with, again, as with gel-based method, with some protein chemistry methods for some historical reasons and to be able to explain how those modern methods actually work. And we'll start with so-called protease profiling. We've already discussed that we need to block proteases from being able to hamper our proteins. They will cleave proteins at some points between some different amino acids. And there are different classes of those proteases. Some of them are really they don't quite care between which amino acid to break this peptide bond, and they will just try to degrade everything down to amino acids. Some of them are quite picky about where to make this cut. For instance, we've already discussed that insulin is actually can be produced by process which requires 4 different proteases. But those proteases are highly specific and they will be able to make those cleavages only for this protein. So we need some protease which can make good enough cuts in our proteins at some certain positions, but it will not make them too rapidly, so they wouldn't degrade our protein just to amino acids. Like in genomics, you like nucleases, which can get you only some cuts at some predefined positions, right? Of course, environment, temperature, pH, and so on can modify your proteases, as well as with nucleases. You need some certain buffer to make those cuts. And you need some concentration, because I think that you know about this star activity of nucleases. If they are at high concentration, they will start to cut your DNA not only at predefined position, but also at some other positions. So the same actually is here. And the one protease which is highly usable and it also really quite nice to have in mass spectrometry is trypsin. we'll discuss why it is important that our uh, fragments after this protein of the work of this protease should have some certain amino acids, because in mass spectrometry only some amino acids can be helpful to ionize your peptide, and some other will not get any charge at all and wouldn't be visible at mass spectrometry equipment. So trypsin is such a happy shot. This protease is really nice because it gets some predefined position. It's really cheap compared to other proteases, and it cuts the protein for such peptides which have a guarantee to have at least one amino acid which can get this charge and can be visible at later stage at mass spectrometry. So, what is good about trypsin? It can cut your big protein to small peptides, and it cut between those two amino acids. And for instance, if you have such a protein, or part of protein actually, then your protease will cut here, 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 here. Of course, we have RKL here, so with two cuts, and there is an end of our protein. As you can see, we get some parts which are not too long, not too short. They are really nice for future investigation by mass spectrometry. But what we can do and what we need to know here is that this protease can be modeled using some computer software, but it's not. It has some. It doesn't make just only one cut at only one peptide bond at a predefined position. There are also some drawbacks. For instance, if we have this cleavage site here, it's quite similar to nucleases. We have some sequence and we have something upstream this sequence, downstream this sequence, and we have this cleavage site here. And we can make such a table of where this protease will make this cut. As you can see, it will make cut if at this position there are either of those amino acids, but it will make cut unless there is a proline in this position. If there is proline, trypsin will not make any cut. It could be used to our advantage. For instance, if we know the sequence partly of this amino of this peptide, and there is no cut just by using trypsin, we know that there is probably proline following those amino acids. So we already have some information. Also, if we know the whole length of our proteins, and now we know the number and the length of those small peptides, we actually already have some data which can be used to search the database, right? Because different proteins will have different primary structure, and the number, the length, and actually those peptides will be different for different proteins. And if we, for instance, need to decide between two proteins which differ enough, we can do this just by trypsinizing them and see what kind of fragments we get from it, right? Also, there is another problem. It will not cut your protein in those conditions. There is different amino acids here, and there is different combination at which this protease doesn't work well. You can get to this URL to see more about specificity of the trypsin, but as you can see in most textbooks, there is an oversimplification. The trypsin always will make this cut between K and R here, and we'll always have this number of trypsin fragments. Not so, actually. As you can see, trypsin is a little bit more sophisticated enzyme. But there is another problem. Since trypsin is a protein, and trypsin works on another proteins by making those cuts, can trypsin actually trypsinize itself?
Looking at this slide, yes.
I
Trypsin
will- yeah,
outlines.
yeah. So the problem is that if you just put trypsin in solution, it will actually make those cuts on itself, and after it will change its specificity. It will be less specific. It will have some activity like a little bit different protease, chemotrypsin, and it will have additional cuts, which is not always what we want. Because of it, we need to buy specialized trypsin, which either have some modification, artificial post-translational modification, which prevents trypsin from self-trypsinization, or what we can also do, we can immobilize this trypsin on some membrane. And why it is beneficial? First of all, if it is immobilized, can it self-cut other trypsins?
No.
No, it's already immobilized. Also, we can make this membrane permeable only for small peptides, but impermeable for full-length proteins. So we can combine proteinase activity and getting these small fragments with filtering, and we'll get only small fragments after this. So it's just an ultracentrifugation vial which have this additional immobilized trypsin on it, and afterwards we can get our small peptides which we can use for mass spectrometry and other ways. Where you can get this information? You can get an overview of immobilized proteases here, and also there are some kits which provide you with buffers and different proteases, because we've so far only addressed trypsin, but there are other different proteases which is usable for proteomics work, like lyc, arc, and so on and so on. We will talk about them a little bit, but it is understandable why we need different sets of proteases. For instance, if our protein have just some small number of sites which can be addressed by trypsin, then we'll get big peptides which are not quite easy to work with, or vice versa. There's a lot K and R in our amino acid sequence, and we'll get just some D peptides or G peptides, which also not quite what we want to identify a protein, because there is a lot of different proteins which have just two different amino acids side by side, right? It will not allow us to search a database. It would be easier for us if we have some fragment of, say, 5 amino acids, 10 amino acids, right? It will allow us to search a database more efficiently, and of course the information which we can get here is bigger. So we have different additional proteases which can help us in those cases. So what we can do without mass spectrometry right now, we can do so-called protease profiling. For instance, you have some protein, and you want to make some educated guess about its primary structure. What amino acids are there, and what are the positions of those amino acids? It's quite similar with what you can do with nucleic acids, with different nucleases. And what can be done is using a library of proteases, or actually also some chemical agents like cyanogen bromide, hydroxylamine, and so on, which also can break some bonds, peptide bonds inside your protein. And afterwards, what you can actually see that this protein is probably not modified, because methionine is the first amino acid, right? We know that there are M in A before, so the second amino acid is A. We have this cut of trypsin here, so there is an R here, and so on. Just one by one we can get some draft of sequence of our protein, right? Of course, it's time consuming and nobody uses it just for sequencing. It is mostly used for confirming some of findings which you already done by other methods. But this protein profiling is usable for instance, getting events of some modifications, because some amino acids which are modified would be acceptable for proteases and they will be not cut in this stand. And there are some other cases where it can be useful. You can get to this URL to see how different proteases can cut your protein of interest. What will be the length of this fragment, what will be the sequence of those fragments, and so on, so on. It will actually, we actually will require this because one of the questions in the upcoming test would be the selection of a protease which will be most beneficial for some certain protein. So you will have some primary structure, and you can get to this site and test different proteases to get, for instance, some fragments which will include some amino acids which have some modification. Because for some reasons, it can be that you will need to not only answer the questions, are there some modifications, but also the position of this modification. And if you will be able to cut the protein in parts, and some of the parts can differ by this modification, you can just compare which part have this modification, either by mass spectrometry or by some protein chemistry methods, and it will allow you to address this question quite easily. You don't need to sequence this protein, right? You only need to have mass of those two peptides and see whether there is such modification or no modification at all. Was it clear? Or at least understandable? Right. I have 6 minutes yet till the break. So, as we've already discussed, just to sum up everything that we've discussed so far, we can use protease profiling to get partial sequence of our protein, or to ask questions where were those post-translation modification on our protein, or confirm if there are some changes or mutations if we, for some reason, unable to sequence our organism. For instance, there is some problem with primers or whatever. We can actually see are there any changes in amino acid level of our peptides, just using such easy to do and not quite costly method as profiling. So, just before the break, first method of identification of our proteins. It has only historical value, but just to start. For instance, if you have some peptide and you want to know which amino acids are here, because if you know the set of amino acids, you can already simplify the task. If there is no proline, there is no need to actually check for it and to try to somehow get other methods to sequence your protein, right? And if your protein have, for instance, only 3 different amino acids, it's easy to sequence it even with protease profiling or some other even simpler methods of protein chemistry. So, what we can do? If we'll put our protein at highly acidic environment of 6 molar hydrochloric acid and boil it for 1 day, what we will have is that all peptide bond will be broken. We'll have only 3 amino acids. But we can use some analytical methods like ion exchange chromatography. There is a strange choice of immobile phase for such a chromatography like polystyrene beads. Polystyrene is what is usually used for plastic, which is used for some tableware and so on. And as a co-elutant, we use sodium citrate because it will try to stick to the same centers as our amino acids, and we can do this just column chromatography. So you have a column of those beads, you pour your sample, add those sodium citrate here, and just make a gravity flow and just collect some fractions. And actually there is some books with some numbers when you know after what time what amino acid will elute from this column. And you can just change your vials or some vessels, and then all you need to do is just to stain it, make some correlative reaction which allows to see whether there is any amino acid in solution. And which is also nice if you have 9-hydrine as a staining agent, then all amino acids except for proline will have this nice violet color, and proline will have this orange color. So you already will be able to actually say which amino acid is here, and also you can use spectrophotometry to get some idea of concentrations. If some amino acid is twice as much by concentration in your solution, then you know that those amino acids, this amino acid repeated twice in your protein or your peptide of interest, right? So just to start, we can get amino acid composition just by using such a technique. So now we'll have 10 minutes break and we'll continue with adenine degradation. If you have any questions, please ask me. If not, 10 minutes free time.
Да, конечно. Я тут сложно просто не забыть по поводу профилирования протеазами. Просто правильно ли я понимаю, что когда мы получаем вот этот вот набор разделенных пептидов, мы это тоже по факту электрофореза разделяем по виду?
Можно электрофорезом разные есть методы. В принципе, можно сделать электрофорез в геле, где очень большая концентрация акриламида и бисакриламида. И в принципе можем различать даже такие маленькие по весу фрагменты. Есть другие методы, монохроматография это сделать. То есть в принципе мы можем, есть много способов, как это можно получить. На самом деле, в некоторых случаях, когда мы не точно можем определить, скажем, последовательность, нас интересует масс-спектрометрически, мы как раз с такими маленькими пептидами лучше работаем, чем большими. То есть что-нибудь от 4 до 12 букв, это вот такое самое для нас удобное. И в принципе, если мы не можем спектрометрически сказать, какая у нас последовательность, у нас нет генома и так далее, мы можем, если у нас есть, скажем, 2-3 варианта, какие нам подходят, подобрать просто протеазу, посмотреть, а где есть разрезанный или нет. Дальше мы спектрометр посмотреть, есть такой фрагмент или нет, и тогда мы уже можем не догадываться, что есть 3 варианта, сказать, что да, здесь вот именно делаем кислотой. Потому что, например, лизин, лицин и залицин, они вообще одинаковый вес имеют. И на спектрометрически их вообще никак не различить. Некоторые пары аминокислот, если они между собой не раскололись, будут иметь очень близкую массу. А метиониноокисленные будут иметь очень близкую массу к другой аминокислоте. То есть масс-спектрометрически, что мы будем говорить, в некоторых случаях масс-спектрометр есть, конечно, методы, как можно пытаться сделать, но вот так вот не может отсеквенировать прямо вот полностью. И тогда придется прибегать к этому старому методу.
То есть, правильно я понимаю, что в таком случае, если мы говорим про то, что вы говорите, когда мы не можем нормально это сделать, то как бы, мы тоже загружаем масс-спектрометр же не целый белок, а тоже уже пептиды. Вот пептиды вот эти. А когда мы в итоге по масс-спектрометру не получаем четкой картинки, то мы возвращаемся.
Прям можем взять другую протеазу.
Просто другую протеазу и как бы делаем разные варианты этого всего.
Ну, у нас там, видите, например, лицин, да, не очень понятно.
Я поняла. Я просто смотрю вот на нижнюю,
да.
как будто бы мы вот эту вот нижнюю строчку загружаем.
Нижняя это
факту,
все, что
да,
порежет. Если мы все смешаем вместе, то у нас будет вот такой набор. То есть в некоторых местах у нас нет протеазы, которая могла бы, скажем, здесь разрезать.
Мгм.
Да. Ну, либо еще пока не нашли. Наверняка в природе, конечно, есть, но вот из тех, которые коммерчески доступны, нет.
Мгм. Хорошо, то есть на спектрометр мы берем только, ну, скорее всего, берем
Какую-то
смесь одну
строчку.
из...
Да, вот триптин у нас вот такие фрагменты даст, например. Аркцеа вот такой набор.
Ну да, понятно.
Там пепсин, ну, пепсин не очень много для спектрометра, очень длинный, куча коротких, конечно, да, так же как СПН не очень. Цианобромид хороший тоже даст, например, на спектрометрии. Вот. А эта ссылка, там можете сюда вставить последовательно, дать как будет
Ну, это-
порезано, просто чтобы это не вручную делать.
Мгм.
Тем более там есть модели, например, для трипсина, РКЦ, которые учитывают вот все эти варианты, где он не будет резать, например.
Мгм. Хорошо. Тоже вроде не сложно. Просто я боюсь, и когда...
Ты обычно когда руками делаешь, начинается самое веселое.
Ну да.
Когда жидкости прозрачные, 2 дня переливаешь, а потом ничего не получается. Где ошибся? Ну, приходят журналисты и говорят, покажите нам что-нибудь красивое, и приходят жидкости красить. Им обязательно нужна синяя перчатка, капающая носиком с какой-нибудь окрашенной жидкостью в гель.
Я как-то делала видео для одной стипендии, ну, и там нужно было показать, чем я занимаюсь. И я просто разводила разные, ну, типа, условно, купрум может дважды, просто чтобы у меня была синенькая водичка красивая.
Ну, мне теперь удобнее, многие метаболиты растений, к счастью, окрашены. Теперь можно даже ничего не придумывать.
Не понимаю.
С мозгами было сложнее, там все было прозрачное.
А вот какими методами, из тех, что вы рассказывали, вы вот, в повседневной практике используете? Просто, как я поняла, вот эту вот идею ионно-обменной хроматографии, она, ну, как вы сказали, это больше историческое значение.
Да, это просто как вариант первый. Но на самом деле, в принципе, белковая химия наверняка где-нибудь используется.
Просто на самом деле я часто слышала про ионно-обменную
Не,
хроматографию, но, возможно, не только в этом
смотрите,
контексте.
это метод, который
Ну да,
позволяет
это
нам
может-
разделить заряженные аналиты.
Мгм.
Поэтому сама по себе ее ионная хроматография очень часто используется, мы еще о ней будем говорить. Но в плане для выяснения аминокислотного состава только в пищевой, господи, как по-русски будет, фуд-индустрии, в общем, когда нужно пищевую,
Называют еще в России.
ну, наверное, так. Когда нужно вот оценить, какие аминокислоты есть вот во всем сырье, тогда да, действительно кипятят с кислотой, но, честно говоря, уже мало кто вот так анализирует. Обычно это все просто в газовый спектрометр и все.
Мгм.
Потому что аминокислоты очень легко делаются деривации их, они становятся летучими. И с газовым спектрометрией гораздо проще работать, чем жидкостной, она дешевле, и их уже много где есть. Вот поэтому то, что я говорю в историческом плане, это именно использование их
Ну да,
для
да.
идентификации, для получения первичной структуры и так далее. А руками, ну смотрите, выделение белков, все, что я рассказывал, это мы всегда используем. Да, одномерный гель, посмотреть вообще, что там выделилось и получилось. Ну, хочешь, не хочешь, тем более сейчас покупать уже немножко проблематично, даже биорат уже не все возят. Делать, да. Двумерные гели, честно говоря, у меня плохо получается делать другой человек. Я не знаю, от чего зависит, по-моему, от фазлона. Те же самые реактивы, все то же самое, но вот у кого-то... Да. То, что я буду говорить про идентификацию при помощи масс-спектрометрии отпечатком пальцев часто, то, что касается фрагментации часто, ну, микроскопии с антителами вообще каждый день практически, ну, вот, в общем, набор. Но это все зависит от объекта, что смотрите. Но это все будет зависеть от объекта, что вы смотрите.
А в смысле от объекта, то есть для растений, условно, для разных типов организмов или для разных
Ну, смотрите.
типов данных?
Если работаете с человеком, у вас уже материал будет фиксирован, потому что медики вообще не дают ничего, и якобы они работают с материалом. Да, им в статье вставляют, что пытаются первым и последним автором просят, чтобы вставляли. Поэтому с медиком не всегда удобно работать с человеческим материалом. С фиксированным что можно делать? Фиксированные белки вы уже на гель нормально не положите. Я его
А
показывал.
фиксированный в смысле
Формалине.
просто
Ну, считается, что материал человеческий, он первично патогенный. То есть они могут выдать в живом состоянии.
Просто я знаю, что вот срезы делают, ну, как-то в парафине или в парафине
Парафор.
Да.
Какая-то формалин и есть. Парафор, но просто формалин
Да.
полимеризованный. Его
Да.
подогреть, он обратно распадется.
Я, значит, что-то про другое. Ну, возможно,
Ну, скажем так, вот,
я с человеком не работаю.
когда я раньше работал, до 2012 года, все было проще. С многими тканями и прочим можно было работать, в общем-то, биологом. И материала было много. С 2012 года приняли часть веселых законов, но тебе вообще не понятно, чем работать. Все должны только медики делать. Медики, естественно, делать это не будут. Поэтому выбирайте то, с чем будете работать, перспективно на будущее, что не запретят.
Я пока что с микробиомом работаю.
Ну, это хорошо, кто же его запретит?
Ну, просто по факту это тоже очень тесно связано с медиками, потому что как раз-таки сбор образцов и...
Ну, сбор образцов, понимаете, микробиом, конечно, может включать, если будете секвенировать там, в общем-то, и ДНК человека, и все. Но давайте подумаем. В принципе, ну хорошо, вот вы будете иметь, скажем, даже полный секвенс какого-то человека. Много ли вы сможете по этому секвенсу сказать? Сможете по секвенсу сказать, например, расу человека?
Кстати, ну, здесь интересный вопрос, потому что если мы говорим...
Если у вас 100 человек...
популяции,
Нет, смотрите, если у вас 100 человек одной расы и 100 человек другой расы, то сравнить вот эти, где у вас 100 человек одной и 100 другой сможете, а если один?
Вот я про это и хочу сказать, что если мы говорим про неафриканские популяции, то, скорее всего, нет. А если мы говорим про африканские популяции, то у меня здесь вопрос, ну, возможно,
Ну, африканские,
вопрос,
да,
потому
можно
что
еще
африканские, они очень варьируются.
Можно говорить еще о айнах или Японии, например, там, да, еще какие-то такие варианты есть. Но
Ну,
это все-таки,
просто-
согласитесь, редкие такие случаи.
И почему, как бы я говорю именно про... Я не уверена, что Айна или Япония в таком случае будут хорошо различаться, только если не по конкретным генам, типа я, по-моему, помню, что у японцев выделили какой-то фермент, который специфичен именно для них, который лучше помогает расщеплять...
Копия этого фермента. Фермент один и тот же. Копия. Оказались.
В смысле копии фермента?
Ну, дупликация.
Просто...
Дупликация немножко изменилась.
Просто больше ферментов или типа вариация какого-то?
Ну, вариация, но опять же, понимаете, там
Ну, вот по
хороший
вариации
секвенс и
можно
все
понять.
остальное. И опять же, где человек сейчас живет, да, то есть сколько можно получить, ведь сейчас вокруг генома идет такая борьба, что это персональные данные, что-то там вот перечь. Не знаю, что моногенные заболевания, я согласен, да, если есть моногенные заболевания, прекрасно, но моногенные заболевания себя настолько часто проявляют. А уж эти идеи о том, что вот мы сейчас отсеквенируем, там, не знаю, какую-то популяцию, сделаем против них биологическое оружие, вообще чушь собачья. Учитывая, как сейчас все перемешанное, так тогда получается половину своей популяции уберется сразу же. Вот. Так что с объектами сейчас стало сложно, с некоторыми. С растениями, к счастью, хорошо. Они это, ни этического комитета не нужно, если не краснокнижные, ничего. Можно килограммы выделять, они не жалуются. С бактериями, правда, еще лучше, но...
Ну, смотря с какими.
Okay. So, shall we continue? Everybody is here, I think. And
Marina should come also, but maybe she's still on her school.
okay, but it is recording our lecture, so just be sure to watch this presentation because there will be a lot of questions. I a little bit edited our test from the last years, so it will include some different questions. So just be sure that you will be able to see the whole presentation.
I will. Our first mate says that the records are very good. So at least previous previous lectures. So.
Fine. So, but you can always ask me on Friday. We will have our semi-practice session, questions and answers session. So just be sure to ask questions here. Oh, now we've already seen that there are some possibilities to get at least partial information about primary sequence of our proteins or peptides, But I've already mentioned that first protein that was sequenced by Mr. Sanger, who is also known for sequencing of nucleic acids. But his method of sequencing was not quite good because it couldn't be easily automated. What we want just to put our sample and get our data, right? We don't want to have a lot of hands-on work and which is not quite repeatable and so on and so on. So we have a little bit different idea of how we can sequence our proteins, and method of adenine degradation, which I will be covering now, was quite popular just I think till the start of this millennium. Nowadays it's also used because not always it is possible to get good sequencing from mass spectrometry data, and for some certain reasons, not all organisms are sequenced, and that's why we can have a need of de novo sequencing some peptide or even protein. How this method works? Actually, the idea is quite simple. The realization of this idea is not so simple. There is so-called phenyl isothiocyanate. Isothiocyanate group is widely used. For instance, you use it to label proteins by NHT group. All the most fluorescent dyes came in this form, and then you can put this dye on your proteins just by mixing those two solutions, having some heat, and this group will covalently bind to NH group, right? But the idea here is this part of this molecule which allows us to cleave here, to cleave this peptide bond. Just changing the pH will result, we have this high or basic pH, we have this reaction at basic pH environment, then we lower pH, and what happens is auto-cleavage of this radical with this part of amino acid from our peptide. Now we have this free and alpha and terminus, so -1 amino acid. And afterward, we can repeat this cycle. We can again rise the pH at a basic level, we can add this chemical, we can have this reaction, we can lower pH, and so what we actually do here is we cleave our protein from N-termini, just one amino acid by one amino acid. Of course, it couldn't be free amino acids, it could be such a modified version, but at low pH, what will also happen is some groups will regroup, some just basic organic chemistry. I'm not a chemist, so I can't actually tell you what happens here and why, but the guesses here is, I think that there is a charge here, and it will regroup in this molecule because we have low pH. Afterward, we can make just chromatography with some detector, and we'll be able to assign each peak on this chromatogram to different derivative of amino acid. So, what we actually do here, we cleave amino acid by amino acid. They are modified in a certain way, but we have a library of retention times of those different amino acid derivatives. And we can compare our chromatograph, our peak, with these peaks of different standards. So this idea is that we can repeat this cycle over and over again, get this chromatography done, and afterwards we can say that, for instance, the first one was methionine or alanine, the next one is another amino acid, another. Was it clear? So the idea is straightforward. The problem here is that this reaction can only occur if there is no water. So we have to buy specialized reagents which are totally dry. We have to use special equipment here to be able to have this reaction. And also, can N-termini of our protein or peptide gets modified in any way?
Can you repeat?
We know about post-translation modification of radicals of our amino acids, but can some post-translational or even spontaneous modification happen at an N-terminus of our protein or our peptide? Can be something that is blocking this NH growth on N-termini.
Probably.
Not probably, there is a lot of such modifications. And if this NH group is not available for this reaction, will we be able to sequence our peptide?
We should remove this modification before somehow and then start to do it.
Yes, if we know that there is such modification.
Yes.
Because if nothing happens, there could be a lot of different reasons, right? It's not only modification. How can we actually get at least some partial sequence of our protein if we know that our N-termini is modified? Cut it by proteases, right? Absolutely. And there is one of drawbacks of this method that you have to be sure that you have this free and alpha N-termini of your protein or peptide. Also, we can do the sequence only from N-termini side. C-termini is not possible to sequence with this chemistry. Actually, nobody was able to devise this kind of chemistry to sequence it from different side. And also, even though that this reaction is really selective and it have really high yield of 99, almost 99%, what do you think? If we'll try to sequence 10 amino acids or 20 amino acids, what will happen with the quality of this sequence? After, let's say, 10, 20 or 50 amino acids.
It would be worse.
Worse, those errors will sum up. If you have so happened that some of this acid wasn't cleaved by the previous cycle, it will cleave at another cycle. And you'll have two peaks on a chromatogram. One will be higher, another one will be a little bit lower, but still, which one of them is true? And then after 10 or 20 cycles, we'll have even more of such errors. So this method also only possible to be used up to 20 or 25 amino acids. So you can sequence, as you can see, it's a real sequencing, your peptide to protein from N-termini with the depths of, let's say, 20 to 25 amino acids. Right. We've already discussed this and how it actually looks. This is the real apparatus. Unfortunately, there is no Edman degradation sequencers in St. Petersburg. As far as I know about two of them in Moscow, one of them at Ruski Island. It's somewhere near Kamchatka. So there are not much of them, but there are three of them in Finland, unfortunately available for us right now. So, if you will ever need it, please ask our Moscow colleagues at Shemyakin Ovchinnikov Institute. They have them. Also, most of this is actually just a chromatography equipment, and this one is what actually makes this cycle happen. Another drawback, it's really slow. You can get one amino acid for about 40 to 45 minutes. So if you want to sequence 10 amino acids, it's already half a day. If you want to sequence 20 of them, it's 1 day, and so on, so on. Also, since it's chromatography detection, you will need a lot of starting material. Mostly, you will need 100 picomole of your protein. Is it a quite a big concentration or a lot of concentration of your protein, or is it actually a lot of material or not so much of material? If we are talking about molar masses here. And let's say that our protein is, let's say, 120. You can calculate as actual weight of a protein, of pure protein. You see that it had to be more than 97% of purity protein of this site at a concentration of 100 picomoles. It's somehow that's milligrams, right? Of course, for smaller peptide, let's say 20 kilodaltons, it's less. For something like 7 kilodaltons, it's even less. But for big proteins, it's a lot of starting material. So actually, we've just summed up all the problems that we have here. And another problem is if we have some certain post-translational modifications. Some of those modifications will not allow this cycle to continue and will stop this modification. Others will cleave this amino acid, but chromatographically it will be another peak. It wouldn't be those standard peaks like here, and you will have some problem of deciphering this information. So I would like to say that for some most frequent modifications, there is some data in database of this equipment which allows you to say that serine was phosphorylated, for instance, and so on. But for most of less frequent modification, there is no such data, and you will have to guess it by yourself. So that's the problems, and we've discussed it. And now we have to continue with mass spectrometry, because mass spectrometry is actually a method of choice, nowadays at least, for proteomics. Of course, mass spectrometry costs a lot, but single analysis on mass spectrometry is not as expensive. You can afford it from laboratory budget without any problem. Also, mass spectrometer can get you information about a lot of proteins, a lot of peptides in a small time. And for some certain cases, you can even use mass spectrometer to make some guesses at a primary sequence of your proteins. Of course, it's not possible to just sequence every peptide that will be analyzed. You will see later why. But even if we have just partial information, we can always search protein or genomic database to get this partial information and to be able to compare it to some known genes or some known proteoforms. It is easier than just to sequence it like it was in N degradation. Have you had any courses on mass spectrometry? So I have to do just some basic introduction on mass spectrometry as well. Well, if we have some molecules in vacuum, can we interact with these molecules by any way? Just a molecule in a vacuum.
So can we put something?
What we need to be able to easily operate with this molecule? By either electric field or magnetic field.
Yes.
Charge, electric charge, right? We can either add electron or get electron, but it is really hard ionization and it will break most of molecules. Or we can add or remove protein from a molecule, which will also get some charge on it, right? So it is there is need of some way to make our specimen charged, but to make this charge in such a way that our protein will not degrade from this action, right? Actually, first mass spectrometers used really harsh condition to ionize any substances, like just using a stream of electrons in vacuum. And as you can imagine, that electrons at energy of, let's say, 50 electron volts is enough to break a lot of covalent bonds. And what we will have in such a spectrometer after ionization is not your molecule of interest with a charge, but some fragments of your molecule And some of those fragments will be charged, some will have no charge. And as we've already discussed, we can't manipulate uncharged species in vacuum. They don't have charge because we can't interact with it either with electric or magnetic field, and they are actually lost for us, right? So the idea is that we have somehow ionize our sample. Also, we somehow have to make separation. And what we are interested in is actually mass of our molecule, but here, since we use charge, we can only access such a characteristic as mass to charge ratio. Because if we have two charges on our sample, it will interact with either electrical or magnetic field more than singly charged species, right? And if we have the same charge, let's say just one charge, but two molecules of different weight, it will also interact a little bit differently with either electrical or magnetic field, right? So, mass spectrometer actually have three different parts. It's parts which will ionize your sample, a part which will make some separation, either in time or in space, or even in frequency domain, and some part which will allow you to detect those charged particles. And of course, just to start, from historical perspective, try to start with some not so good an example for proteomics, but a good example to start understanding mass spectrometry as electron impact ionization mass spectrometer. This uses just a beam of electrons, and our sample have to be in a gaseous phase, so it have to be evaporated in some gas. We have some sample here, some molecule, and gas impacted by electron. This energy is enough to fragment actually this molecule mass spec. Of course, there is a small chance that we'll get a molecule with, let's say, positive or negative charge. But mostly what we'll have some parts of this molecule with either charge, right? Afterward, what we can do, we can separate those fragments by mass to charge ratio, and what we'll get actually is what you usually have at gaseous mass spectrometry equipment. Let's say you have some small molecule, let it be something like this. I would be picturing hydrogens here, because it's easier. And if we have such a molecule, we'll probably have some mass to charge ratio, which will characterize this molecule. But mostly, what we'll have a series of fragments all over our chart. Here is intensity, here is a mass to charge ratio, and since our molecule have symmetry and those parts of molecules are the same, what you will see in the mass spectrum is those groups which also looks the same, right? Because we can get here, 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 here. It can be fragmented either way, and we'll get a lot of different parts from a molecule, and we can plot it like this. For proteins, what do you think? Will it be beneficial to have such fragmentation in our source?
It is very small fragmentation.
Yeah, as you can see, for our, let's say,
But like the two amino acids, really.
for this chemical, we get something like this.
Protein should have.
What will end up? A lot of different peaks tightly packed together. Of course, for, let's say, just free amino acid or deep peptide, it can provide a lot of information, because it can be used to get information about even some molecules which have the same elemental composition but different isomerization, because this picture will be a little bit different. But it will be hard to interpret. What we actually want from our spectrometer is the ability to make this part as soft as possible, to ionize the whole protein, or at least the whole peptide, and get, in ideal condition, just one charge, either negative or positive. For proteins and peptides, we mostly get positive charge. It's easier to interpret it, and historically it was the first ionization condition to get just those positive species. But actually we can get both. We can analyze even at even with negative charge, but it's some specialized case. So, how can we separate our masses and charges to get this nice graph? There are different ways to do those. This so-called quadruple mass spectrometers, ion trap mass spectrometers, Fourier transform mass spectrometers, whatever, cyclotron mass spectrometers. I will be talking about them just at a basic level, because we don't quite need all those technical details. But what we need is what kind of mass spectrometers are actually needed for proteomics, and what each different type can help us with, and what can be drawbacks and actually advantages of different types. So I will be covering them just superficially. First type to be described is actually quite an easy idea. For instance, if you have some means of ionization of our analyte, which will provide us with just single charge ions. Let be mass of our ion plus proton, and it will also have a positive charge. As you can see, we don't have a mass of our analyte. It is added with a mass of just one proton, so it will be 1 Dalton more than what we expected. And what we can do actually, let's say this one have Z is 1, so this mass in Dalton. Say that it is here. And let's say we have another molecule of, let's say, 210 MZ. Since our charge will be also just 1, we can rewrite it as Dalton's. It's a big one with the same charge. And what if after the ionization we'll provide our ions with a small push by electric field? Let's say that we have some electrode with some free part here, because if it will be just a part of leaf of, let's say, metal plate, we'll just accelerate our ions and they will be just be buried inside this thing. So let's say that we have some net of small wires, and what we can do is to apply a lot of negative charge here. Since they are positively charged, they will be attracted here, right? But we can switch off this charge at some time. What will happen? Since there was charge when they start to move here, and the charge was extinguished by the time they get there, what will happen to the trajectory of those two particles? Will they continue to move in a vacuum in this direction?
Will be change charge.
We've applied negative charge for just some time. For instance, here's some time, and we've applied our negative charge sound. Just no charge, light it, turn it off.
So and there it starts, this part starts to be just
They-
positive and maybe so, I mean not
Negative, positive, so
Yes.
it will attract. Then we switch it off, and it will continue to.
it starts to be neutral without any
Why?
charge. I
This
mean,
is one, yeah. The net, yeah. But those charges.
yes, it has charge, so it can.
Yeah, absolutely. We just pushed it a little bit. And since there is a vacuum and their mass is negligible compared to the gravitation of our planet, they will continue to move forward. But since we've provided each of these particles with the same amount of impulse, what would be the speed of those two
The
particles?
smaller will go fast.
The smaller will go faster, right? This one will go a little bit slower. And then?
May I ask?
Yeah,
why?
absolutely.
It looks to me if we have some powers of gravity, in this case, do we have it?
Yeah, absolutely. But
So the like the bigger one should be faster, no?
gravity is here. And
Ah,
it is so
okay.
small compared to other forces that we can just ignore it.
Okay.
This force of, let's say we have here 5 kilovolts of extraction, and here we have just the hole is just 1 centimeter. So we have 5 kilovolts per centimeter of electric field. Compared to our gravity, it's nothing.
Okay.
It will just shoot in this direction with really high speed, right? But what if then we'll allow them to move some distance, and here put some, let's be red one, some detector, which will only detect cases when this charged particle hits the detector. The smaller one will be the first one to get the detector, right? And if we'll continue to count time here, this would be our smaller 110 Dalton. And our second one would be a little slower, and it would be somewhere here. And it would be some microseconds, for instance. So, actually, now we can recalibrate our time scale to mass to charge ratio, right? And if we have some known standards, we can say that, let's say, it's some ballpark, of course, let's say 50 milliseconds is 110, and let's say 98 microseconds is this opposite. Was this clear? Are there any questions? What is not quite clear is that actually for most of ionization sources we can get this situation where we can get doubly charged species. And also it can be either double charge, but it can be plus 2 hydrogen with 2 plus here. And if this happens, what we will have in our spectrum is what would be
faster
even faster. It would be somewhere here.
and correspond to another molecule weight.
Absolutely. So that's why we actually using not Dalton here, but M over Z. We can have one-to-one correlation only if this only single charged species, if there is more than one charge. Because it's not. So actually it's not a Dalton, but M over Z. So what we've just described right now is so-called time of light analysis. So what we analyzed is time which is needed to get those ions to our detector. And some of mass spectrometers will have in their naming something like something TOF, time of flight, something. For instance, there is such a way of ionizing analytes as electrospray ionization, ASI. And if we have such kind of analyzer, we'll call our mass spectrometer like ASI, TOF, MS. There can be a lot of different ways, but as you can see here, the naming is actually came from those three parts of our mass spectrometer. So, let's continue. I just wonder what sound is internal or external. I was afraid that something is with the computer because it's also have some noise. So, even if our mass spectrometer working fine, still there will be some limitations. For instance, the resolution. We've put here just one single bar on our graph for one single ion. But actually we accumulate a lot of ions to get a picture, and what we mostly will have is something like this. It is an intensity, so the number of ions, and here it will be even more. And it will never be small enough, but the smaller will be the peak, I mean the sharper it will be, the higher will be the resolution of our mass spectrometer. Of course, the higher the resolution, the easier will be for us to identify those peptides, identify our proteins. So it's really nice to have high resolution. And how can we describe this resolution in some terms? If we zoom in on this part of the graph, what we'll see is that we have some distribution which have Gaussian form. And for Gaussian, we can get half of the width. So that is the width of it, and this one half of it, and measure what is this problem. So you probably came across this idea of full width, half maxima, and some different courses as well, right? So the idea is that we can get perfect resolution. If we get some resolution from our mass spectrometer, it can be modeled as this Gaussian distribution. For Gaussian distribution, to just have an idea how big it is, we can get just some number because it's up to infinity here. It will be coming closer and closer, but if we'll have infinite times, we'll still have some of cases here and here, right? And as we do in statistics or in physics, we have somehow to describe it, how we can do it. For instance, we can get the whole height of this peak, get half of it, and at the half of this peak, get the numbers here from what number to what number in m over z, Daltons or whatever this peak actually covers. And for instance, here we have quite a good resolution. As you can see, there is almost 500, and the log power of this mass spectrometer can be easily calculated using this formula, and as you can see, it's quite high. We'll need this resolving power because resolving power is not homogeneous on this scale of m over z. For instance, if we have something really light, we'll have this kind of distribution. It's also Gaussian distribution, but it will be more product, right? That is why describing this resolving power of mass spectrometer, we need to be sure of what kind of analytes are we working with. For instance, this 5,000 of resolving power is enough for working with small peptides because they have relatively small mass. They will be finally resolved down to isotopic level. But if we are working with some protein of, let's say, 120 kilodaltons, those peaks will be wider and wider. And to resolve two proteins which differ by, let's say, even 10 daltons will be impossible at this range with the same equipment, which is high, which we thought about resolving power. Was it clear? The resolution is different at different masses. That's why we actually put in this formula this m here, m over dm. From this idea, we know that if we are working with some small molecules, we can resolve them quite nicely. And if we are working with some big molecules, the resolution would be not as good. That's why actually mostly in proteomics we are working with peptides. They're small enough. Or that's why we can work with multiply charged species. If we get, let's say, 100 charges on a protein, even if this protein is 100 kilodaltons, it will be visible somewhere here. The resolution is more or less good enough. So, was this idea understandable? The resolution is not homogeneous, and we want to work as this low part of m over z, if it is possible. So let's continue then. There are different types of mass spectrometry equipment, and as you can see, resolving power is also different. For small peptides, quadruple is enough. Of course, it's not quite good for proteomics, but quadruple can be used. If we are working with peptides and want to get small differences, for instance, we have oxidized methionine and phenylalanine, which have almost the same composition. To have a difference between them, we'll need something of this kind of resolution. And if you want to get just fine structure of isotopic distribution, we'll need either Orbitrap or FT-CRMS with really high resolving power. So isotrap mostly not usable for us. Quadruple is only for some cases, and time of flight or other mass spectrometers are the most widespread. Actually, there are two types of mass spectrometer which are mostly used in proteomics, in screening proteomics. It's time of flight mass spectrometers and Orbitrap mass spectrometers. I will be describing them a little bit in more detail. Also, why this resolution is actually important for us. There is some protein, bradykinin, really small protein, I would say that it's a peptide, and what you can get from this protein mass spectrometry at different resolution is those two pictures. What do you think? Why there is such a nice picture which have half a Dalton ratio between peaks? What can cause such a picture? It's just one single protein or peptide, it's small enough, and we have a number of peaks. And the distance between those peaks is half a Dalton. What can we propose? Why this is
Like one protein fragment.
It's not fragmented, it's still in one piece.
Additional charges.
Charges, yes, right, but why this half Dalton? or if we'll have singly charged specimen, it will be 1 Dalton. Why we have this strange picture? What kind of isotopes of carbon we have here on planet Earth? And what is the distribution between those two different isotopes stable?
14.
And not 14. 14 is actually decays quite rapidly. 12 and 13, right?
Yeah.
And 13. Dating by different isotopes is possible because some of them have different half-life and so on. We have about 1% of isotope which have almost 1 Dalton difference, heavier than our standard C12. And what we have here is actually this isotopic distribution. Let us see why it is the case and what we can actually get from this picture. Why it can be important for us. Let's say that we have just some number of carbons. I will omit hydrogens for the sake of understanding. Of course, there's 4 valences, but still. If I have some ratio between light isotope, stable isotope, and heavy isotope, then we can say that if we have, let's say, have ratio 1 to 99, which is not quite the true for carbon, but still for the demonstration purposes. If I have 100 carbons, what is the mathematical expectation of having about one of them, heavy one? Let's say we have C12, 12, 12.
0.001. No, 0.1.
There is 100 of them, and 1 of 100 in our nature is, what is mathematical expectation?
Expectation.
Expectation.
Probability.
It's the same as we have a coin and we toss it just 2 times. What is
50%.
1?
Here 1%. 1.
Here's 1, right?
It's
Absolutely. So for such a big molecule, it's more likely that we have at least 1 carbon-13, right? And if we have some molecule and have some mass spectrometry here, if it is short molecule, then there is some only C12, which is the most prominent. We can have occasionally some heavier isotopes, but they would be almost invisible, right? Then we have some molecule, we have more carbons. For instance, let's continue here. And it can be that the case of having at least 1 C13 is the same as having all C12, right? And what will happen next?
But 2.
More C13.
Yes.
So on, so on, so on, right? That's the pictures which you can get from proteins, because proteins have a lot of carbons. Of course, we omit such problems like we have even different isotopes of hydrogen and some other isotopes, but mostly it's a carbon. Also, we have different ways to calculate the mass of our ion. For instance, if you have some chemical, organic chemical, you will have so-called molecular mass. What is this mass? Is it mass of only C12? In this case, what would be probably the molecular mass? We can just get something like this, and this would be our molecular mass, right? Because there is a ratio of isotopes of carbon here. the smaller the molecule, the closer would be this weight to so-called monoisotopic weight. What is monoisotopic weight? It's the weight of your compound of the most frequently found isotope. For carbon, it's C12. So for this peptide, monoisotopic mass would be this mass, and molar mass would be this mass. Was it clear? Because if we have really poor resolution, just look at the top graph here. Can we get here monoisotopic mass from such a poor resolution?
Yes.
How? And from the bottom one, here, can we get monoisotopic mass if you have this green band resolution?
If there is the C4, yes.
Only here molar mass, right? And here is our monoisotopic mass, which is just hiding here. Right? So, the idea, and take a message from this, what we want from mass spectrometry. One peptide, one peak. What we actually get. And also, we only put here singly charged peptides. And what if we have doubly charged peptides? Let's try to see what will happen. First of all, if I have two charge, so mass to charge ratio would be lower for them. So for this one it would be somewhere here. For this one it would be somewhere here. And for this one it would be, I think, about here. But what about the difference between those peaks? Here we have just about one Dalton, or even we can say Dalton because it would be multi. So 1 m over z. And here, since we have double charge, what do you think about the distance between them?
Distance inside,
Yes, between those two peaks, for instance.
it starts to be bigger.
Bigger or smaller?
Small.
This
M over Z. And is this bigger now? This is 2. Here's 1. Right? And if we have triply charged,
There will be overlapping.
they will be overlapping, and also what will happen with those distances? What would be
Small.
this one will overlap, unfortunately.
Also.
Right? So what we want? One peptide, one peak in Dalton. What we actually get? Series of different peaks from the same compound. Right? If you have different compounds, those peaks can even overlap. That's why actually mass spectrometry is not so straightforward. And that's why we have different ways of ionizations, different mathematical apparatus to get rid of those problems. But if we have, let's say, such a picture and see this fine structure of isotopic distribution and see that the difference is 0.33 Dalton, can we say what was the charge on this particular substance? Yeah, absolutely. Here, just half, so it would be, and here we have just 1, so it would be, right? So this structure can already help us to simplify what we have. If we have such numbers, we can say that, well, this is triply charged, this is two charge. There is also, let's say, two charge, and somewhere here is one charge. And we can actually use this information to group those peaks. We can see that this, and this is the same ion. We can just sum it up, right? If we have high enough resolution. But what if the resolution is not so nice? And what we have actually is this. Can we somehow get an idea of a charge of such a compound?
Yes,
Absolutely right. How can we do this?
if the space is wider.
Absolutely, but the space is different, right? We have 3 and 2, and we have to calculate this distance. And for instance, we know that we have some ions which are, let's say, M plus 3 hydrogen, 3 plus here, M plus 2 hydrogen, plus 2 here. Please take into consideration that we have not only the different charge, but the mass will be different. We have additional protons here, right? But can we mathematically, knowing this difference and not knowing this and this, calculate having only this distance?
Can you repeat?
For instance, let's presume that we have this poor resolution and can get this isotopic distribution which can easily tell us what the charge ratio of our molecule. And we want to know mass in Daltons, not some m over z, but the mass of our peptide, for instance, or protein even. And what we are bound to is just using some, let's say, quadruple mass spectrometer with resolution of, let's say, 2000, and what we'll get from this mass spectrometer is something like this. Probably here. As you can see, the distance is different, and we know that it's only just one specimen here, just one peptide, and we have a lot of peaks. And we can calculate distance here, or here, or here. Can we, just having those distances, or just one distance, get an idea, actually two distances I needed, of either charge of each of those peaks or even molecular weight? What do you think?
Yes.
It is possible, absolutely. But what would be easier? Just counting how many those isotopic peaks are here, or doing some complex math? Of course, that's why we actually, another reason why we want high resolution mass spectrometers. And actually, how can a spectrometer of a mixture of 3 different peptides from the same protein look like? As you can see, there is a lot of information. There are some fragmentation because there is no such thing as ionization without any fragmentation. Some of molecules can be not stable enough that will fragment by themselves. There are some additional charge states. That's the real story. Only 3, as you can see, peptides are here. Let's see what we can do. First of all, as you can see here, even there is nothing, there is some peaks, but should we concern every single mass as a peak because there is some signal?
No.
No. We have to define such a characteristic as a noise. And this noise is the floor above which, actually 3 times above which, we can count peaks. Below those 3 times the noise, there is no need to even concern yourself about any peaks. It would be just noise or not resolved at all. What I would like you to do is to see this YouTube video where there are some explanation about this noise, but to be honest, actually there is no one single standardized way to calculate this noise. Different algorithms, and as you can see, noise level is a little bit different. Each software for each mass spectrometer will get you a little bit different results. So that's just a problem. Actually, there would be a lot of things that would be non-standardized. For instance, resolution can be as a resolution itself or as a resolving power. And those two used interchangeably, but they have a little bit different formula to calculate. Just one example. Also, what we can do? We can have an idea that this noise level is not the same at different masses. As here, you see that at a lower mass to charge ratio, there is a lot of noise. Here, noise is less, and we can subtract this background, this noise, just to have this baseline. As you can see, after the subtraction, we have this nice picture, which is already quite better. And here we can then, having the noise level, get information about those peaks. Usually, we do not store the whole spectrum because there is a lot of additional information. For instance, you have a spectrum from, let's say, 10 m/z to 10,000 m/z with resolution of not 0.001 m/z. The size of a file on a computer would be big enough to have half a CD disk, right? And it's only one spectrum. Do you need all this information? Or you can just extract peaks from this list, right? And it would contain just some numbers which already have an impact on the file size. And it is easier to process. So this is called peaking, and the blue line is so-called profile of your mass spectrum, and red lines are so-called centroids. They already mark your peak, the center of your peak actually. Was this clear? In proteomics, we're mostly interested in use this centroided data. If you are working with metabolomics though, you are most interest to be able to see this raw data, because sometimes it is possible that in some of these cases there is another peak which is not visible, right? And picking software wasn't unable to get it, but doing the same thing manually, you can annotate this peak and use it for some cases. So, profile, centroid, and also our mass spectrometers can have some error in mass. We need to calibrate them. Why this error is there? For instance, we are talking about time-of-flight mass spectrometers. We have this length where our ions have to cross without any field. So we just push them and then continue to flight. What can happen with the length of this metal tube if we change temperature in our room? Can this metal tube get shorter or bigger if we, let's say, have lower or higher temperature?
It can deform.
We can get this type of error. Even if our mass spectrometer is fully calibrated, absolutely operational, you just open a window and have this problem. Actually, the funny thing is that you can just yell at your spectrometer and it will also get you different results. There is a YouTube video of the mass spectrometer getting the same readings, and someone just yelled at it. And
It
it just
mass spectrometer had a soul and it was offended by the yelling.
It's solid, but There is some models of elasticity even in metal parts, and we are measuring nanoseconds and micrometers actually to get this information. So don't yell your mass spectrometer, it can just spoil your data. Funny, but there is such a video. So we have to somehow get an information on how good is behaved our mass spectrometer. What is the error here? And it can be expressed either in milli or microdaltons, if we are not happy enough, or what is more preferable is to measure it using this formula, and you will get results in point per million ppms. So mostly the error of mass detection in ppms, not in resultant power, because it will be different for different masses, but it is down converted to those ppms. The lower the ppms, the better. 10 ppms is quite enough for our proteomics work. If you can get to the mass spectrometers 10 ppm uncertainty in mass detection, it's what you need, nothing more. It would be already enough. So, we've already discussed the idea of different masses. There is so-called molar or average mass. There is so-called monoisotopic mass, and there is also one mass which, fortunately, is not used in proteomics, but unfortunately used in metabolomics, is so-called nominal mass. Do you know what nominal mass is? For instance, if you have some element, let's be oxygen, does it have just some number of Dalton, or is it some number dot something?
Number dot something.
Why this dot something? If you have neutrons, positrons, electrons, and we know the number of them, but if we'll combine them, this mass doesn't sum up. What happened? Why we have this difference? We've just summed all the masses of all the particles and get a little bit different result. What is it? Mathematics is wrong. Or what are we missing here? And also, if you'll try to get this nominal mass for different elements, you will see that for lighter elements there is higher mass. And after iron, there is also some difference in mass which gets more and more. And iron have the less possible mass if you just sum all the neutrons, protons, electrons. Alright, if we, let's say, have uranium, what will happen with this uranium at some...
With radiation, like it will become much less.
It will, and also some energy will be released, right? You will have, let's say, tritium and deuterium, and you push them close together. What will happen with them? Our sun is actually an example.
Hydrogen and...
It will fuse, and also some energy will come out. But if you get some iron, you can squeeze it, you can do whatever with it. No energy will be added even if you push it together, or if you try to just disassemble it to some particles. So-called mass defect. Some kind of mass is converted to this energy of forces which hold together the nucleus of these elements, right? This mass defect. Actually, why it can be important for our proteomics study, and where can we use it, and why high-resolution instruments can be of use here. Is it that only carbon have isotope, or other elements also can have stable or long-lived isotopes as well? Nitrogen, oxygen, nitrogen. And also, one element which is
Sulfur.
sulfur, right, but hydrogen, right? And if you will be able to zoom here at this isotopic distribution, for instance, just here, what you will actually see at really, really high quality, high resolving power, and so on, so on, so on, mass spectrometer, is that, yeah, you have those C12, 1, C13, and so on, so on. But there will be also some additional peaks. Like here we have 1 hydrogen, 2, and so on, so on, so on. So just having all this information, if it would be possible to just get all this information down to the highest resolution possible, you will be able just using this information to get Bruta formula of your compound of interest, right? So this mass defect is also quite interesting. Unfortunately, it's also only possible on the high-end instruments like cyclotron Fourier transform mass spectrometers, which I think 2 of them are in Moscow and 1 of them is in Novosibirsk, none of them in St. Petersburg, unfortunately. That's a pity. So it is possible to do. Okay, but down to nominal mass. Nominal mass is if we have this mass of, let's say, 12, 0, or something, just average to this part. And what is interesting, we will sum up all of our elemental composition using only this part of mass. This would be a nominal mass. It could be a little bit higher than molar mass, or monoisotopic a little bit lower. It was used in old era when mass spectrometry had really low resolution, and this was called unit resolutions, because the unit was 1 Dalton at the time. Now we're using more advanced mass spectrometers. So, what we've covered today so far is that there are different masses, and we somehow need to know which masses we are working with, because monoisotopic masses are one thing, especially for heavy peptides. Molar mass is could be used in chemistry and some lower resolution mass spectrometry, and there is also a nominal mass. So just don't mix them up; they are absolutely different. Also, we've talked about one type of mass spectrometry equipment, time-of-flight mass spectrometer. We've been able to see that it is possible that just one substance will give a rise to a number of different peaks, and we know how to get some information from this picture of different peaks, like charge state, for instance, either by these clusters or by this monoisotopic distribution. And tomorrow we will be talking about how we can mathematically get all this information from those numbers, and what are the different types of mass spectrometer, and what are the advantages and disadvantages to proteomics. Also, we'll discuss in more details ways of ionizing our substances so softly that only molecular ion, so the whole protein or the whole peptide, will be ionized without any, at least we hope so, without any fragmentation. Since today the lecture was a little bit, I think, difficult for you to understand, I will permit myself to end it 10 minutes earlier, and tomorrow we'll continue with our mass spectrometry. As always, questions welcome, so ask any questions.
If there is any standard library that is associated with mass spectrometry that could determine which protein it is?
You can make such a library yourself. If you have, for instance, transcriptomic database, you can convert it in proteins, because those triplets can allow you to get primary sequence of your protein. Then you can use this peptide cutter. Let me just get it here. Peptide cutter, like, yeah, here. To cut all your in silico generated proteins in parts, and here's your library. But usually we do not have this library for mass spectrometry for proteomics. We have some search engines and programs which don't rely on this library but do all what we've already talked about inside their programs. So they get some FASTA file, either protein or transcriptome or genome, whatever, and they inside them in silico translate them into nucleic acids and uses some model of what what peptides of trypsinization can be available, and then compare those masses to those masses that was in silico derived. Was it clear or something?
Yes.
We will be discussing it in depth next lecture, actually, so it's just a brief overview.
Thank you.
You're welcome. All right, then.